Let's start our lesson. Our first slide is about centrifugation theory. So, what is centrifugation? Centrifugation is a technique used for the separation of particles from a solution of any type, according to their size, according to their shape, their density, their viscosity, and then the rotor speed. So, the particles are suspended in a liquid medium and placed in a centrifuge tube. What is centrifuge? Centrifuge is a type of machine that helps in centrifugation. The tube is then placed in a rotor and spun at a definite speed. Got it? The particles are suspended in a liquid medium, placed in a centrifuge tube, and then the tube is then placed in a rotor and spun at a definite speed. Separation through sedimentation could be done naturally with the earth gravity, as it would take ages, but centrifugation is making that natural process much faster. Rotation of the rotor about a centrifuge in a central axis generates a centrifugal force about the particles in the suspension. So as the particles are placed in a centrifuge tube and the tube is then placed in a rotor and spun at a definite speed, so the rotation of that rotor creates a centrifugal force upon the particles in the suspension, and then the centrifugation process starts. So all the work is done by centrifugal force, which is generated by the rotation of the rotor within the tube that creates the particles in the suspension to be separate from one another. So let's talk about the factors which have an influence on centrifugation technique. Those factors are density of both samples and the solution, the temperature, the distance of the particles, and the rotation speed. All these factors have somehow an influence on centrifugation process. Okay, so what is centrifuge? As we have studied earlier about the centrifuge machine, so what is a centrifuge? Centrifuge is a device that separates particles from a solution through the use of a rotor. What is a rotor? Rotor is something like a motor which spins at a definite speed in a circular motion. So according to the use of the rotor, centrifuge device separates the particles from a solution. The particles may be simple cells, subcellular organelles, the large molecules, or all of which refer to here as particles. Centrifuge procedures. If you are talking about the procedures of the centrifugation process, then we have mainly two types of centrifuge procedures. One is known as the preparative procedure and the other is analytical. Preparative procedure is done to isolate the specific particles, while the analytical procedure involves the measuring of the physical properties of the sedimenting particles. This is the difference between the two procedures, which is preparative and analytical procedure. Centrifuge method. As a rotor spins in a centrifuge, a centrifugal force is applied to each particle in the sample. We have studied in the first slide. Then the particle will then sediment at the rate that is proportional to the centrifugal force applied to it. The viscosity of the sample solution and the physical properties of the particles also affect the sedimentation rate of each particle. At a fixed centrifugal force and a liquid viscosity, the sedimentation rate of a particle is proportional to its size, and the difference between the particles' density and the density of the solution. These are all of the factors which have an influence on the centrifugation force. We had already studied in the previous slide. Okay, come to the next slide. Here is the diagram. Diagram shows before centrifugation phase and after centrifugation phase. Look at the second tube after centrifugation phase. It's so clear and smoothly as compared to the first tube, which have some suspended particles, suspended debris, and suspended all the other suspended materials which have to be eliminated from the solution or from the liquid. Solution is of any type here. Solution is not specific in the centrifugation technique, as centrifugation is about to clear out or clean out all the debris or the suspended unwanted particles from a liquid. So solution is of any type here. In a solution, particles whose density is higher than that of the solvent sinks, and particles that are lighter then it float to the top. So here is the particles which have higher weight sinks at the bottom, and the particles that are lighter float at the top of the liquid. This is all about the factor density. So here is the influence of the density on the centrifugation. If the density of the particle is higher than that of the solvent means any solution or any liquid, then the particles sediment or sink at the bottom of the solution. And if the density is lower than that of the solvent, the particles float at the top of the solution. So the greater the difference in density, the faster they move. Clear? Here is another diagram, rapidly rotating rotor. Here is the diagram of or a sketch of the rotor, which is inside in the centrifugation machine called as a centrifuge, used for separating the material by applying the centrifugal force on the particles of the. Look at the diagram. Before, the particles suspended in the liquid, and after, the particles are sedimented at the corner or at the bottom of the solution, and the liquid is cleaned out. To take advantage of even a very tiny difference in a density to separate various particles in a solution, gravity can be replaced with the much more powerful centrifugation force provided by the centrifuge bottom of the form. By using the centrifuge machine for the centrifugation process instead of the gravity centrifugation, which was an earlier or which was a natural process of centrifugation, the gravity can be replaced with much powerful force, which is also known as the centrifugation force or the centrifugal force, which is applied on the particles of the suspension by moving the rotor at a definite speed, and which will help out the particles to be removed or to be suspended at the top of the bottom and to be cleared out from the solution, and the solution becomes then smoothly and clear and clean for use. So centrifugation is basically a technique which is used to remove all the unwanted particles, all the unwanted substances from a solution or from a liquid. Solution may be of many types. There is no any specific solution. Solution may be of some sugar. Solution may be of some even juices. Solution may be of some any material, any type of solution. If you want to clear out any type of solution and to clean out all the debris, all the suspended particles from that solution, you have to perform the centrifugation technique by the use of the centrifuge. And centrifuge works by applying the centrifugal force on the particles of the solution by spinning the rotor at a definite speed, which creates the centrifugal force. And due to that centrifugal force, the particles will be separated out from the solution either by sinking at the bottom or by floating at the top of the solution. So here centrifuge, we have already studied about the centrifuge. So we have centrifuge types There are following types of centrifuge machine: microcentrifuge, small benchtop centrifuge, super speed centrifuge with a super speed rotor, and the general purpose centrifuge, which is used only for specific purposes or for specific solutions in biology, and the ultracentrifuge, which have a much more higher speed than that of all other types of centrifuge. So this is all about our centrifugation technique. Inshallah, we will study the centrifuge types further in our next lesson. Thank you so much. Allah Hafiz.